variation. So, I think it's interesting to start this out by talking about the odds that, because you guys know if you have a sibling, you might look a little bit like them, right? But I want to know the odds that you and your sibling would be twins, meaning that you have identical DNA. You would obviously not be the same age, right? Unless you were like actual twins that are formed in a totally different way, right? They're formed by the splitting of zygote, right? And so I want to know that the odds of two siblings that were not born at the same time being twins, right? So let's go through and let's crunch the numbers and see if this could happen, what the odds are of this happening, if it's ever happened before. Okay? So there's two laws that we have to look at that are going to affect this. The first one is called the law of independent assortment. And what this law says is that the way that the tetrads are going to orient themselves, and so this always happens in prophase 1 when the tetrads form. So we'll say in prophase 1, when tetrads form, the orientation of one tetrad has no effect on the orientation of the rest of the tetrads. Okay? So, in order to tackle this, we're going to look at a very simple cell. We're going to look at a diploid cell that has 2n of 4. Okay, so it's got a diploid number of 4. All right? So the way that these could orient themselves then, if the diploid number was 4, is they could have, we're going to do one long chromosome, one short chromosome again, just because it's easy to sort of denote them that way. So long chromosome, short chromosome. Okay, and then we're going to do blue and red. Okay, so here's your tetrads. And so the first possibility is that they orient themselves with the colors. And obviously your chromosomes don't have colors, but we're just denoting that they're different homologous chromosomes. Maybe one from your father that could be the blue one, and one from your mother that's the red one, right? They've oriented themselves so that the colors are on the same side. So blue to the left, red to the right, okay? So on this one, it's going to be different. So the long chromosome blue will be to the left, long chromosome red to be to the right. But then down below, you've got the short chromosome red on the left, and the short chromosome blue on the right. Okay? These are the two possible orientations. And what the law of independent assortment says is that, let's say that the first tetrad orients itself like this, with the blue on the right and the red on the left. That has no bearing, no effect at all on the way that the second tetrad orients itself. It can orient itself like this, or it can orient itself like this, and it's not going to matter. Okay? It doesn't have any effect, or the first chromosome doesn't have any effect on the next set of chromosomes. Okay? So these cells are going to go through the process of meiosis 1. And when they do that, when they go through meiosis 1, they are going to create haploid cells. Because we know that in meiosis 1, what happens is the tetrads rather, whoopsies. The tetrads are separated and the sister chromatids remain connected. Okay, so now we've got 4 possible orientations here, 4 possible outcomes. Okay, and I want to denote that these are sort of separate instances here, so I'm going to draw a little dotted line down the middle here to represent that these are cells that are all on a little unique path. They're not connected. Okay? Okay, so like I said, tetrads are going to be separated here, so we're going to end up with this side, the long blue, and the short blue, and this side has the long red and the short red, and this one's going to be mixed. So the left one has the long blue and the short red, and the right one has the long red and the short blue. Right? Okay, so then next, each of these, first of all, we've already gone into a haploid state here, right? Our 2n was 4, so we could count 4 chromosomes up here, but now we only have 2 chromosomes, and that's important that in meiosis 1, after meiosis 1, we're now going to be n is equal to 2. We're haploid cells. So next, let's go through and let's put our 4 possible cells down here. Copy, paste, paste, and I didn't leave myself enough room. Paste. Okay, so those 4 cells are like this. This process represents meiosis 2. So this is meiosis 2. And in meiosis 2, that's the one that's just like mitosis, we said. And so that's the one that's going to pull the sister chromatids apart. Okay, so in this one, we're going to end up with long and short of blue, and this one long and short of blue. That one, both reds. This one's mixed, so the long ones are blue, and the short ones are red. And this one, the long one is red, and the short ones are blue. So we've essentially created 4 types of cells here. And each of these types of cells is going to be viable because they each contain one of each homologous chromosome. We said that the homologous chromosomes were either short or long. Right? And so we can note those as L and S, right? So we'll say that that L equals the long chromosome. And S is the short chromosome. And so you know that in order for this to be able to live, it needs to have one S and one L, right, at least. So we'd say that this is L and S, this is L and S, this is L and S, and this is L and S. So they all have one long and one short, right? But now we need to denote which ones they have. So this first one has the blue L and the blue S. The second one has the red L and the red S. The third one has the blue L and the red S. And the fourth one has the red L and the blue S. Okay, so these are what we call genotypes here. We've kind of shown which chromosomes have gotten segregated for each of these. Does everybody agree that there are 4 possible outcomes here? We started with 4 chromosomes, and we've made 4 unique cell types. Okay, so that might seem like it's totally doable, but the problem is that since each of these can segregate into one of 2 cells at the very beginning, that means that the math behind this is going to be exponential, right? So it's going to change exponentially based on the number of chromosomes. The math behind this looks like this. So math for independent assortment. Okay, so the first thing that we're going to have is a 2. And that 2 represents the fact that in meiosis 1, it's going to be separated into one of the 2 sides because the cell's going to split into 2, right? It's either going to go to the left side or the right side. Okay? Then we need to know how many times that has to happen. In this case, it happened twice because there were 2 homologous pairs of chromosomes. One of them had to separate here, and then the other one had to separate. And so that gave us this, okay, where this number up here was equal to the haploid number, okay? Which would be N, right? That's then going to equal 2 to the 2 is what? What's 2 to the 2?
4.
It's 4. Okay? And 4 then is equal to the number of possible outcomes. Okay, so if the haploid number is 2, then the number of possible outcomes is 4. And that's simple, right? But consider humans. Humans, we have a haploid number of 23, right? So we have to do 2 to the 23. So 2 to the 23rd is 8.4 million, approximately, right? Okay, that means for every unique person, their gametes, their sex cells, have 8.4 million different combinations of chromosomes that they could have just based on the law of independent assortment. Okay, so to start that out, okay, we talked about what are the odds that you look just like one of your siblings and you're not actual twins? 8.4 million means that there's a 1 in 8.4 million chance that you will have identical genes from one of your parents. Okay? Now that has to happen twice because both the sperm and the egg have to be identical then. So you take 1 in 8.4 million and you multiply by 1 in 8.4 million and it becomes 1 in 16 million. No, wait. You multiply the 8.4 millions together. So it's 8, yes. 8.4. Oopsies, I should just do this. Times 8. 1, 2, 3. Yeah, 1 in 7.05 times 10 to the 13th. That you would look just like a sibling. But that's not even the half of it. That's not even close. Okay? It's going to be way more than that. And the reason it's going to be way more than that is because this isn't even the source of most of the genetic variation. Source of most of the genetic variation comes from a recombination that happens during prophase 1 that's called crossing over. Law of crossing over says this. It says that during prophase 1, these things form that are called tetrads. Okay? So during prophase 1, homologous chromosomes physically cross over one another to form tetrads, which your book calls bivalents. Okay, and when they do that, the site that they cross over causes a exchange of genetic information. So we can say that genetic information is exchanged at the sites of crossing over. Okay, the site of crossing over has a fancy name. It's called the chiasmata. What's it?
We're trying to figure out how to pronounce that.
Chiasmata. Okay, so the chiasmata is the site that the actual crossing over is happening, right? And then the process of exchanging the genetic information is called synapsis, right? You could also just call it crossing over. Nobody's going to get mad at you for that. It's never going to get points off on an exam or anything like that. But the technical term for the process is called synapsis, right? Okay, so let's look at what this does. Here is a chromosome, and I'm drawing it like this on purpose. So I'm drawing this chromosome as a bunch of sort of squiggly lines. And the reason for that is that don't forget that a chromosome is actually a molecule of DNA, a very long strand of DNA that's wound up a bunch, right? So this is one chromosome. After S phase, you would expect there to be another one that is identical to it, and they will be attached in the center, just called the centromere. Again, because this was created in S phase, we would expect these two sister chromatids here to have identical DNA to one another, right? Okay. Now, in a diploid organism, you've got two of these things. You should expect them to be roughly the same size and the same shape-ish. It won't be perfect, but it'll be close. With the exception of the 23rd chromosome that has X and Y. The Y is sort of like this little stubby thing, and the X chromosome is a normal size, right? So this is prior to prophase 1. They're just hanging out, okay? Now during prophase 1, they're going to cross over. And when they cross over, there's going to be a physical location in which they are going to overlap each other. As you'll learn a little bit later on, there are areas of the chromosome that are non-coding DNA, right? We call these introns, and then there are areas that are coding DNA, and we call those exons. They specifically code for protein or something like that. Okay. It turns out that the introns are really important, the non-coding regions, because those are sites where crossing over is going to occur. It's the beginnings and the ends are going to happen in introns. This means that if the genetic information that gets crossed over isn't spliced perfectly, exactly on the same nucleotide, then it won't damage the gene because it's non-coding portions. And so you don't normally see a whole lot of messing up of genes when they cross over like this. This is also showing it a little bit disingenuously because this is showing just one crossing over location, right? There's nothing to say that it can only happen in one spot. right? These two chromosomes here could cross over in 10 spots, right? They could cross over in 2 spots. These chromosomes in the middle could cross over in 2 spots, and the ones on the outside could cross over in 1 spot, right? There's almost no limitation here as to how many times they can cross over and as to which ones need to cross over. This is just showing it in the very simplest way where they're only crossing over in 1 spot, okay? The result of this is the exchange of genetic information. So after they get separated then, in this case, we've got one of the sister chromatids that's totally the same as it used to be. And the other one now has a spot on it where it's been crossed over, so it's got a red spot on it or a spot that contains DNA from the other chromosome, from the homologous chromosome. Okay, then this one is again going to be mostly red, but it's got the identical spot crossed over. Like this, and then this one didn't cross over at all in our example. So, let's go ahead and try to do some calculations here. In order to do this calculation, you would need to know the number of crossing over locations per chromosome, which is going to be a number in the millions, right? Then you would need to know the number of possible times that they could cross over, which I said could be anywhere from 1 time crossing over to maybe 100 times crossing over. So we multiply 1,000,000 times 100, right? Then we multiply that by the number of chromosomes, which we said was 23, right? Then we multiply that by this original number that we got, which was 7.05 times 10 to the 13, and we end up with a number that's like in the 10 to the 25 region. Yeah?
How'd you get the 1 million to begin with?
The number of spots, the number of possible locations on each chromosome that could cross over. Now, it could be a little bit more than that because there's probably more than 1,000,000 genes on each one. But by all accounts, we don't think that each gene is capable of crossing over independently. Some of them are inherently linked. Okay? So, yeah. What are the odds then? If it's up in the 10 to the 26th or 10 to the 25, have there ever been that many people on Earth? Will there ever be that many people on Earth, even if you consider all of them? No. Not even close. Right? We're at 7 billion right now. To make that jump from 7 billion to 10 to the 25th is astronomically different. Right? So, will a person ever look like their sibling if they're not twins? You might look similar, but will you ever be identical? No. It's impossible. Not impossible, but statistically impossible. You have, almost as much statistical chance of taking a pencil and dropping it and having it fall through the desk because all the atoms are aligned. Right? It's probably not going to happen. Right? So, questions on that? That's why we're unique. That's why we're also unique, even your siblings, because of these 2 laws.